Alright, say question 1A, yeah. So we got a chromatography situation, yeah. We got a starting line there. We got serine unknown amino acid. We need to find the RF of the unknown amino acid and compare it there, yeah. So take the distance from the thingy, from the starting line, yeah, to the center of the unknown amino acid. Take the starting line to the solvent front, and then divide them together, yeah. When you do that, you'll find that it's 27 over 80, yeah, which is going to be 0.34. So therefore, the RF is going to be 0.34. And if you compare that to the table, yeah, that's going to be glycine, yeah. So therefore, glycine is the one that you want. So glycine is the identity, yeah. Calm. The amino acids cannot be seen as they move during the experiment. State how amino acids can be made visible at the end of the experiment. State why each. Okay, cool. So the way they can be made visible at the end of the experiment, you need to know this, yeah. So you use a UV lamp, yeah. So use a UV lamp, yeah. Alright, safe. State why each amino acid has a different RF value, yeah? So the reason why they got a different RF value is obviously because you gotta state two things here, yeah? Here there's only one mark, but you gotta state two things, yeah? State each amino acid. If you write that, yeah, you're guaranteed to get the full mark, yeah? So each amino acid has a different solubility, obviously, in the solvent phase, yeah? So it's gonna dissolve differently. That's what's gonna push it further or stay there. Also, they're gonna stick to the stationary phase differently, yeah? That's called affinity, yeah? So therefore, it's gonna stick to the solvent phase in different kind of ways, to the stationary phase in different ways, yeah? And hence, that's obviously gonna be, yeah, you get it. That's also gonna affect how far it moves, yeah? With structure of a Switzer ion, yeah. So a Switzer ion, yeah, is just it's just like a positive NH3 group and it's negative COO group with no other bullshit around it. So this one's got a positive here, it needs to be neutral, it can't be. This one's got negative, it needs to be neutral. This one's positive, can't be. So it's obviously D, yeah. This is a perfect Switzer ion, yeah. You need positive there, negative there, and then no charges and anything else, just straight standard amino. You get me? You also can't have amino on the NH2 group either. I mean, plus on NH2, yeah. Which row shows a pair of bases that can link two strands of DNA with three. So the ones that link are AT and CG. You know that if you do biology, AT is double bond. This is triple bond, fam. You know that from like year 12. So therefore, C is the correct one. Cytosine guanine, yeah. Calm. Alright. Alright, question 4, yeah. Alright, let's get it, bro. Come on, come on. Alright. Draw a circle around the chiral center, yeah. So a chiral center is a carbon that's attached to like four other groups, yeah. So obviously it's this one over here. So that's a CO, CH3, CH, and NH. Alright, calm. And also H. Identify the functional groups here. So we got N substituted amine here, yeah. Amide. So we got C, N, H here, O. And on top of that, we also got the, so that's like a carboxylic acid amide kind of situation. That's obviously amide group. And then we also got that attached to it, R group attached to it. So that's amide. Then you got amine right here as well. So that's going to be amine group as well. Ester and ketone, you don't have that. Alright, so if trichloroane, yeah, completely hydrolyzed in the human body to give a mixture of products here. Draw the structures of two organic products here formed in the complete hydrolysis of trichloroane in acidic conditions. Alright, draw this shit out. Draw the trichloroane here. So give it to you in the previous thing here. So just draw it out again like that. Again, I've repeated this bare times here. If you want to get like, if you want the A star, you gotta get gritty, bro. You get me? You gotta start drawing shit. Alright, calm. So NH here. C C H H. Like, trust me, I used to be a brother who didn't draw nothing out. You get me? Just straight raw dogged here. But eventually, you know what I'm saying? I had to mature. And trust me, it's levels, it's levels to this shit. Like, you get me? Alright, cool. So draw this shit out, yeah. Alright, safe. So we got the ring there, we got the situation, I'm pretty sure that's it, yeah. Alright, cool. So what's this gonna hydrolyze into? Well, obviously we're gonna be breaking the amine bond there, the amino kind of bond here with the NC. That's gonna this is obviously a condensation reaction, so it's because hydrolysis here, yeah. So we're gonna release water, yeah. Or we're gonna use water to break this, but what I mean is that water would have been released when these two were made. So one of these needs an OH group and one of these needs a H group. Obviously this is gonna attach to the H. This would have originally been OH, yeah. So that's the two products right there. So that's gonna be OH, so CO like that, HO. and then the rest of the shit. So CH3. I'm not gonna draw that out fully. CH3 and then H and then N H C H C H. Alright, calm. And then the other one is gonna be this group here with an extra H on that, so that's gonna be just the this thing sort of like that. Attached to the NH2. However, that has a lone pair on it, yeah, and because hydrolysis is done under acidic conditions, it's gonna also be protonated, yeah, by another hydrogen. So therefore H plus is gonna attach to that. So it's gonna be H and then it's gonna lose lone pair, so it's gonna be positive, yeah. You need to include that to get the 3 marks, yeah. That's why they're giving you so many marks, fam, you get me? So that's the answer there, that's the full 3 marks. Alright, calm. It's going to be protonated, yeah. Alright, so if question D, yeah, explain why a structural feature of this enzyme enables it to catalyze. So whenever you're talking about enzyme catalyzing something, not catalyzing the other thing, yeah, it's because it has like an active site, yeah, and that active site has like a specific shape to it, and so it needs to fit into that shape, yeah. If it doesn't fit into that shape, it's not going to work, yeah, and we call that complementary in it. So therefore, I can say that enzyme has an active site. E, an enantiomer is complementary to the active site. And you can say, whereas the other one is not, but I don't think you even need to write that for the 2 marks here. They just allow that. In the biology, you would have had to write the other one is not or some bullshit. Alright, cool. Question 5 is the same question as earlier, I'm pretty sure, but even if it's not, yeah, again, Zwitter ion, yeah, has to be positive and negative and neutral, and the other shit has to be neutral. So it's obviously D, yeah, it's perfect Zwitter ion here, just positive, negative, nice. The rest of the amino acid is nice and neutral, yeah. Alright, so calm. Let's go, let's go. This question is about TLC, yeah? Alright, so it's just a suitable reagent for the hydrolysis of a protein, yeah, obviously concentrated HCl. You need that. So yeah. Alright, calm. So just how positions of amino acids on the TLC plate were located, obviously using UV light. It's protein, so UV light. Amino acid, yes. There's usually a minimum number of amino acids present in the original mixture. So you go up here, you find the minimum, fam. What you got here is you got a solvent moving from the bottom up here, and you got a solvent moving from left, right, yeah. So starting from there, one's moving that way, one's moving that way. So essentially, yeah, what you can do is here minimum we've got one, two, three. But in the second phase in solvent two, yeah, we've got one, two, three, four, five, six, seven. So therefore the actual minimum is gonna be seven, yeah, not three. Because you know, you do whatever is the highest minimum, you get me? Yeah. So therefore it's gonna be seven, yeah. Alright, safe. So just why it was necessary to use two different solvents, yeah? Because some of the amino acids may have the same RF value in solvent in the first solvent, yeah. Yeah, so basically, yeah, in the first solvent, yeah, some of the R values might have been the same, so they're going to appear at the same point, so you're not going to see them, yeah. However, with solvent 2, it's going to have to change the RF value. It's going to be forced to, bro. You get it? Because it's a different solvent, so that's going to allow you to see it. You get me? So all of these ones that are there in solvent 1, they would be just on the same place. That's why you can't see them. You get me? Question 7, yeah. Draw all the hydrogen bonds between complementary. So here we got there's only 2 that can form there. There's only 3 that can form there. So it's bait what this is. That's AT. That's CG. Yeah, calm fam. So just draw OH here. Draw the hydrogen bond like that. 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 Yeah. Calm. Draw a ring around each of the component parts that make up cytosine. Yeah, so that's A, T, C, G, because C and G have 3 bonds. So if this is CG, the 3 component parts is the phosphate head. It's gonna be the hexyl sugar, yeah, and then the this thing here, the actual base here. So that's the nucleotide here. Alright, so state the meaning of the term complementary, yeah. So complementary, all they want you to say is basically that A goes with T and C goes with G, yeah. But you can write it like this. Say the strands have base have bases that match A to T and C to G. Yeah. Come. Say for question 8, yeah, which structure shows a part of a peptide link here? So that's obviously D. D is standard peptide link here, CONH. The reason why it's not like B, obviously the other two don't even have nitrogen. The reason why it's not B is because that's going to be like a urea kind of derivative or some shit. That's going to be like something else because we've got like a CO group attached to that. So it's not like a direct link, whereas N would be attached to another C or some shit. You get what I'm saying? Alright, cool. Question 9, yeah. Two strands of DNA are linked together, whatever. Alright, cool. AG doesn't even bond, CT doesn't even bond, CG bonds, AD bonds, CG is obviously 3, AT is meant to be 2, fam. So therefore that's incorrect. Alright, cool. Question 10. We got use the data booklet to help you answer this question. Say nothing, bro. I don't even need a data booklet, fam. But obviously, you get me. It's okay, yeah. But basically, the reason you need data booklet here is you need to know the formula for aspartic acid here, which if you use the data formula, that's gonna be CH, CH2, COOH, and COOH, yeah. And then NH2N, yeah. Alright, safe. So now if you want to use logic here, you'll realize that if it's pH is 14, yeah, there's gonna be a low concentration of hydrogen, obviously, because you've got a high pH here. High pH obviously means that it's very basic conditions. So what's gonna happen is the acid is gonna try to lower the pH here, and to lower the pH, obviously, it needs to donate a H plus here to act as an acid here. So create the environment to have more hydrogens in it, have more protons here. So therefore, it's gonna donate H from here. It's gonna become negative. Donate H from here is gonna become negative because I have two COOH groups, which means they can both do that. And so hence, it's obviously gonna be D, because none of the other ones have that. Why the fuck would there be NH3 plus? Do you know what I'm trying to say? Like, it's just bait. Like, when you get to a certain level, like, you can just see the bullshit that comes through. Do you know what I'm saying? Like, uh, question 11A, yeah. Complete table 4 to show the sequence of bases that are in the correct order. Yeah, obviously, that's gonna be for 1, yeah. So A bonds to T, yeah. C bonds to G, G bonds to C, T bonds to A, C bonds to G, yeah. Alright, so deduce the total number of hydrogen bonds formed between the 5 bases here. So A, that's going to be 2. So you do 2 plus 3 plus 3 plus 2 plus 3. Yeah, you're going to get 13. Calm. So 13 is the answer. So a base is part of a nucleotide. DNA is trying to show, and you've got base A. You need to draw the one that it bonds to. The ones that it bonds to is going to be no. You just need to draw A. I'm trying shit. You just need to draw the nucleotide here. Calm. So for the nucleotide, the acid, the base part of it, yeah, the acid part is going to be here. This is going to be H, H, H. This is going to be H. This is going to be O, and then that's going to bond onto the next part to form the full head here, phosphate head here. That's H. This is going to be CH2, and that C is going to be bonded here to O. I'm writing it a bit dead. It should be like a little complex thing above it here. I should have written it below. It's calm. And then P, and then double bond O. That's the phosphate part, and then O. Or like that to connect to the next phosphate part here. And then P here is gonna be O, and then this needs negative on it, yeah, because single dash here. Alright, come on. Question 12 here. So this is the same as the question we just did, yeah, where it was aspartic acid in pH 14, so there's no difference. It's gonna be D again, yeah. Again, the same reason, yeah. You go to the formula booklet, draw aspartic acid, yeah, it's gonna have COOH and COOH, it's gonna lose one of the H, lose one of the H, it's gonna be negative, negative there. Because obviously it's high pH, so it's gonna need to donate it to lower the pH here. Alright, come on. Use a data booklet to help you answer these questions. A deoxyribose molecule in a strand of DNA is shown. Name the type of group attached to two deoxyribose at positions X and Y. Yeah? So at X, that's going to be the base, and at Y, that's going to be the phosphate. It's pretty obvious, bro. I don't know what to tell you, bro. It's literally in the previous thing as well. Calm. Remember, the phosphate is going to come from above and below, yeah? So it's going to come from above the DNA strand. That's also the phosphate backbone, yeah? And also below to create that phosphate backbone still is going to be the phosphate too, yeah? Calm. In double helix, adenine is linked by hydrogen bonds to the molecule on the other strand. Complete the diagram below to show the other molecule and the hydrogen bonds between it and adenine, yeah? Okay, safe. So because adenine is going to bond to thymine, yeah, so it's going to be T. So you look at the data booklet and you see thymine, yeah, and you need to write that out, yeah? So I think that's the situation. I think it's in the data booklet. I'm not sure though. Might not be. Pretty sure it is though. I don't think they expect you to know the whole structure, but the structure looks like this. H. So this should be in the data booklet, yeah? It's a bit peak, fam. Listen, I'm gonna draw it. Like, it's gonna look dumb. Because I kind of messed up. So that's gonna cause a hydrogen bond between the N and the H there, yeah. And then here, where the O should be, it will be like that. So the O, and that O is gonna bond to the H there. You get. So that's the situation. I know I drew it like scuffed here, but his ways. But yeah, that's the situation there. Alright, question 14A, yeah. Draw the structure of the nucleotide that contains guanine, yeah. So look at the formula booklet, yeah. You'll see the guanine structure. So you can just draw guanine, yeah, and then draw the nucleotide. So first, yeah, what I would do, yeah, is draw the pentose sugar, yeah. Then and then draw guanine, yeah. I'm pretty sure it gives it to you in the formula booklet, so it's gonna look like that. Come. And then here O, and then that is gonna link to the phosphate above. You need to draw that as a link because the full they're talking about the full nucleotide here. And then from here you're gonna link that to the other phosphate head, so P00 like that, and then O, and then again that's gonna link to the bottom, and then here is gonna be O minus here. Alright, calm. That's what they're looking for. So this the pentose and this, the whole left hand side you should be able to do from like your own, like from your mind. You get me? The right hand part, I think, is a formula booklet. Alright, calm. Question B, yeah. Two complementary strands of DNA from a double helix is in which one strand is attracted. Okay, cool. So we've got a free situation here. So this is CG, so you need to draw G. So what you need to do is the same thing, so guanine kind of structure. So again, what I would say is, do you see the O here? That's going to form hydrogen bond with H, and then you can draw the rest in it. So N, H, and then just draw the guanine structure like that. And it's going to form hydrogen bond with H. And then O like that. Again, hydrogen bond with H, and then yeah, and then just complete it. So N, that, 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 that. Don't know what that shit is. That it's gonna go down again. It's gonna combine with other shit. So you need to make sure you have that. Just mirror like mirror that. You get me? Cool fam. What's going on here, bro? Someone chat to me. Someone chat to me. Someone chat. That's it. Yeah, that's the end. Alright, calm. Yeah, that's it then. Yeah. Alright, safe. Yeah, that's it. State how the interactions in AD differ from those obviously in AT. Yeah, it's gonna be there are only two bonds here. So there are only two hydrogen. Adenine thymine base pair. Yeah. Safe D. Yeah. Urea has a displayed formula, so just why urea is effective at separating the complementary strands in DNA. So instead of those complementary strands that in DNA, they're obviously connected by the hydrogen bonds here. So what they're looking for here is for you to basically say that urea can effectively bond to those can also form hydrogen bond. Basically, that's what it wants you to say. Yeah. So you see this NH2, obviously that's going to be polar. So therefore, that's going to form a hydrogen bond with the O. or whatever instead of the bases, yeah. So the NH2 in urea can form, yeah, H bonds, hydrogen bonds here between urea and DNA. It's trans. Safe. Question 15, yeah. Which structure shows the 2-amino-butanoic acid as a bitter ion, yeah? So obviously that's gonna be D. That's supposed to be D. I'm trying complete shit. I was thinking what? I misread it. It's B obviously, yeah. I don't know what the fuck I was thinking with D. It's obviously B, yeah. The reason for this is it's 2-amino-butanoic, so that means that it can only be a certain number of these. It can only be B, C, or D. C isn't even a Zwitter ion, and D you got a positive one O and you got a negative one NH3. So obviously it's B, bro. Come on, yeah. Let's keep it serious here. Do you know what I'm saying? Name the type of protein structure. So this is alpha helix. Alpha helix is secondary, bro. So alpha helix or beta pleated sheet is secondary. You only get tertiary when you start mixing the shit, and then quaternary is when you do like further holding and shit. Explain the origin of the interaction represented by the dotted is I can't by the dotted lines in the figure above. So you're talking about these hydrogen bonds here, so that's going to link to an O and O whatever and a H, I'm pretty sure. So the reason for that is obviously the bonds are polar, bro. So like the bonds here are polar, this NH is polar, and this CO is obviously polar, so that's going to create polar delta negative there and delta positive here. So obviously that's you get me. Yeah. What more is there to really say, bro? Do you know what I'm saying? What more is there to really say? Nitrogen and oxygen here are very electronegative here, and hence NH and C double bond O are polar, which forms a hydrogen bond between O and H. You also, because it's 4 marks or something, yes, you also got to add like, hence a lone pair. You also got to say that the lone pair is that the lone pair on oxygen here is going to be attracted in it. You can say if something attaches to the H here, that's what's going to form the hydrogen bond, or you can say to the partial charge, the partial charge, yeah, on hydrogen. So obviously it's attracted to the delta positive on hydrogen. Alright, safe. Alright. Draw a circle around each of the asymmetric carbon atoms, yeah? Listen, bro. Um, Asymmetric is the same as chiral, yeah? So therefore it's gonna be this one here, because that's attached to 4 different groups here. This is also attached to 4 different groups, because you got H on that side and OH, that's 2 different ones here, and then also here as well. That's the different chiral centers here. Alright, so if draw the sweater ion of glycine, yeah. So glycine is gonna be CCO, like that situation there. If you wanna draw the sweater ion, yeah, so just draw original glycine. So take that and add the OH that would have been there, yeah, that got bonded to the end. So draw that. So that's CCOH H2N. And now just think about it, bro. Zeta ion, that means that this is gonna dilapidate. That's gonna make that H3 plus, and then so that's the sweet ion there. It's just gonna be a H3M plus here, CHH, COO minus here. Alright, calm. Draw the structure of the species formed when glycine reacts with excess of bromoethane, yeah? So draw glycine again. So glycine, draw that. That's gonna be H2M, CH2, COOH here. And that's reacting with CH3Br, yeah? So this is gonna be nucleophilic substitution, yeah? So it's just gonna be one of the H's here on the amine group here is gonna get substituted with CH3. So you're gonna get CH3 bonded to NH, bonded to CH2, bonded to COOH here, and then plus HBr here, because the it's swapped here, so that's swapped with the H here. Alright, calm. Now you're gonna take that again and react it again with the excess here. So it's gonna keep reacting because it's excess here. So therefore it's gonna be H3 NHCH2. So I'm just repeating what I wrote here. And then react that again with CH3Br. CH3Br. And again, that's gonna react to make that's again gonna substitute the other H for another CH3. So, and the H is going to swap with CH3Br, yeah? So it's going to be CH3, two of those now on the end, and then the rest of it, yeah? So CH2, CH, I'm tweaking, CH2COOH, yeah? Alright, calm. Don't know why it's red now, doesn't matter, bro. Alright, calm. And then that's plus HBr, yeah? Okay, safe. And now what you want to do is you want to react it with it again, yeah? So again, react it, yeah? So CH3 is going to form, so that was tertiary, yeah? Now it's going to form the quaternary, yeah? So CH3 2N, CH2, COOH, yeah? Is going to now react with my man again, so CH3 because excess CH3Br, yeah? And that's going to make, so again, the CH3 is going to add on to that, yeah, to make CH whatever, and it's going to leave a Br minus here this time, yeah? So you're going to methylate it, yeah, to become CH3, CH3, three of those here, yeah? And then nitrogen is going to lose the lone pair that it has to become positive, yeah? So that's going to be CH3, and it's going to leave you with a quaternary ion, yeah? Amine ion, yeah? So CH3, 3N plus, yeah? CH2, COOH, plus Br minus here. And that's the final answer there, except this is just the answer. So just write that, and that's the answer there. Alright, safe. So that's the end of the answer. Deduce the UPAC name of furine, yeah? Alright, calm. So let's look at this furine situation. So it's over here. Threonine, yeah? It's bait, yeah? Obviously that was OH here, and that was another H over there before that. So it's obvious what this is. It's just gonna be butanoic acid, yeah, because 1, 2, 3, 4, yeah. So butanoic acid. The acid is here because that would have been OH attached there before it bonded to N, and this would have been H here attached there before it bonded. So therefore it's gonna be 1, 2. So on the second carbon we've got an amino group NH2. So that's gonna be 2 amino butanoic acid. But obviously we also got on the third carbon here. We've got a hydroxy group as well. So therefore it's gonna be yeah, you get the whole point, bro. It's gonna be 2 amino 3 hydroxy butanoic acid, yeah. Alright, calm. You can put butan 1-oic acid, but it's just butanoic acid in mono if they allow that. Alright, calm. Draw the structure of the species, yeah, formed by lysine at a low pH here. So again, so let's look at lysine, yeah. So lysine is gonna be this shit here on the right hand side. So the only thing missing here is the hydrogen that would have been there on the NH before it bonded, yeah. So we've got hydrogen there at safe, and then that's just lysine, yeah. So now lysine is gonna form the Zwitter ion or whatever. No, it's at low pH, yeah. At low pH is gonna act as a base. It's gonna accept a H plus, yeah, because at low pH is going to be acidic conditions, so there's going to be a surplus of H plus in the environment, so it's going to take H plus here. So it's going to take H plus here because lone pair, that's going to become H, and N is going to become positive. So it's going to be NH3 here with positive because there's lone pair on the N, which is going to get donated to accept the H plus, like how amines are, bro. And then also here, you got the NH2 doing the same shit, so it's also going to be H3 plus here. So just write that now. So H3N plus, so the rest remains the same, and then this becomes NH3 plus. There you go. Alright, come on. Alright. State the name of the sugar, yeah, molecule that is attached to C, yeah, at X, yeah. So at X, that's gonna just go to another one of these, yeah. So that's gonna be a pentyl sugar, yeah, which is 2-deoxyribose, yeah. You need to just know that. So deoxyribose, yeah. And the deoxy part, yeah, the part that's lacking the oxygen, yeah. What it means is it means ribose, yeah, without the oxygen, yeah. So it has one less oxygen than ribose, yeah. And where that oxygen is, yeah, is on the second group in it. That's why it's 2-deoxyribose, yeah. Alright, safe. So just which of the bases A or B forms a pair with guanine, yeah. Obviously A, on the left hand side here, guanine needs 3 bonds here, so obviously A can supply those 3 hydrogen bonds, whereas B cannot, yeah. So therefore I can write base A, yeah. You can say the top NH forms hydrogen bonds with the lone pair. And oxygen and guanine, yeah. Then you can say that the lone pair on nitrogen. Okay. Calm. So those have lone pairs are going to form three hydrogen bonds with guanine, yeah? With the lone pairs on guanine, the NH, yeah? Safe. Cisplatin works, yeah? Because one of the atoms is whatever, bro. Draw a ring about around an atom that in guanine that's likely to bond to platinum, yeah? So that's obviously going to be the nitrogen, yeah? So you just need to circle one of the nitrogens with the lone pair, so it's just that. Safe. An adverse effect of cisplatin is that it also prevents normal healthy cells. See one way in which it can be administered. So if it's also affecting normal cells, yeah, then just use it in small doses, yeah. So use in very small amounts, yeah, and that's gonna help counteract that, yeah. So question 19, yeah, a peptide is hydrolyzed to whatever. For each of these parts, you need to look at the bold parts here and talk about whether it's essential and justify your answer, yeah. Uh, so basically, yeah, wearing plastic gloves here is essential, yeah, because it prevents contamination, yeah. Safe. Now look at the second part, yeah. A depth of not more than that, yeah. It's also gonna be essential, yeah. So if it's too deep here, it will dissolve the mixture from the plate, yeah. Yeah, so dissolve the mixture from the plate, yeah. Allowing the solvent to go to the top here is not essential, yeah, as the RF value can be calculated even if it doesn't reach the top here. Bro, my voice is bare dry. Uh, what's the last part? In a fume cupboard, yeah. Yeah, it's essential, yeah, because it's toxic here. Calm. Part B, yeah, outline the steps needed to locate the position of the amino acids here. So obviously you need to use UV light here on the amino acids here to locate them. So use the UV light and then measure the distance of the spots here of the amino acids here from the initial pencil line here, and then measure the distance of the solvent front from the initial pencil line. Alright, save, and then RF is gonna be distance of spots here divided by distance of solvent here. Alright, come. Explain why different amino acids have different RF values. Obviously, the different amino acids are gonna have different solubilities here. They have different polarities here in the bonds here, so they have different polarities, hence different solubilities in the solvent, and also different affinities here to the station phase. So you need to mention both of them here, the affinities or the retention here, or solubilities here. Question 20. Which molecule requires the design of an optically active drug here? So optically active, bro, is just another word for chiral center. So just which one has a chiral center here? Obviously that's gonna be B, not B. What am I talking about? C, because C is bonded to 4 different groups here. CH, that H is technically above, so like H, CH3, COOH, and OH. That's 4 different groups here, so it's a chiral center here. So therefore, it's gonna be optically active here. Safe. That's it.
To my dead friends, now catch me slipping, motherfuckers tripping. So I do it for my town, how I get down. So I do it like my town, and off the loud, I'm straight, off the lean.